Hello, Diane. Thank you very much for taking part. I think the first question we've got to ask you is, what makes your research important?
Okay, so I thought this was quite a hard question, actually, because what's important to me is not necessarily important to other people. So I thought I should start by putting it into some sort of context, what the area of my research is and where it sits. And I've been involved for more years than I'd like to say, actually, in the study of a particular superfamily of enzymes. They're called a superfamily of enzymes because it's such an enormous family. It's thousands of different proteins, and it's not just because I think they're super, which actually I do. They're known as the cytochrome P450, or CYPs for short. And it's been a particular CYP called CYP51 that's been a particular interest to me over the years. It's present in some bacteria, it's present in fungi, it's present in animals like ourselves, and in plants. And what's really interesting to me is how it's related in structure and function. So what the enzyme does, what the protein does, across these different kingdoms of life. In our own cells, in humans, it's part of a pathway that leads to the synthesis of cholesterol. This is the main sterol in our cell membranes. And in fungi, it's involved in the pathway that makes ergosterol, which is the main sterol in fungi. And so the sterol sits in the cell membrane, and it gives the membrane structure. And it is acts a bit like a skeleton, really, and you get lots of proteins and other stuff intermingled with them, but it sort of forms like a nice structure there in the membrane. And it's been shown that the CYP51 enzyme is essential for life, and as such, it's proved to be a useful target for antifungal drugs and antifungals used in crop protection. And over the years, my research has focused on that subtle difference between human, plant, and fungal CYP51s. So that, for instance, if a fungus causes disease, either in a human or in a plant cell, it's possible to kill the fungus and not the patient or the crop. Really important. And the group has developed an experimental approach to exploit those differences and to test new antifungal drugs and new antifungals against crops. What's become more obvious in recent years is that resistance to both antifungal drugs and agricultural antifungals is on the increase. So much as you see for the increase in antibiotic resistance in the clinic and in veterinary practice, we're now seeing that rise in antifungal resistance, both in the clinic and out in the field. So there's a real need to discover new active compounds. And one way to do this, and the way we've been doing it within the lab, is to understand the differences between this specific fungal drug target, CYP51, and the host cell, either a human or a plant cell. So that's what makes my research important, or at least interesting to me.
It sounds both interesting and very important. I suppose, looking more narrowly now, what was the highlight of the year in your research?
Again, this past year has been very different for all of us, and so for much of it we haven't been in the lab, and so it's difficult to pinpoint a particular highlight. What I have done is sort of tried to think more widely and go back over my own research career, which started in Swansea many moons ago as a PhD student. And I was interested even then in P450 and trying to measure that in the yeast Saccharomyces cerevisiae. So this is brewer's or baker's yeast, but it's used as a model organism in science because of its similarity, not least with our own cells. And I remember it was in the early hours of one morning when I actually was successful in measuring cytochrome P450 for the first time in Swansea. This was after several all-night stints in the lab, and I was very excited by this. So that was one of the highlights, and really sort of kept me interested in P450. But secondly was meeting one of my heroes of P450 research. I was sat alongside a Japanese scientist called Yuzo Yoshida in Graham Richards' lab in Oxford. And both of us were watching the 3D structure of CYP51, this our favorite enzyme in the world, unfold on a computer screen. And I couldn't believe I was sat next to this eminent scientist, and we were both seeing for the same time this structure unfold on the computer screen. It was really quite it was an exciting moment for me, not least I think because back in those days it was very difficult to visualize protein structures, so that was really quite exciting for me. Since then, and in the past couple of years, I've been working alongside Dr. Claire Simons in Cardiff University. She's in the pharmacy department there, and she's trying to develop new antifungal drugs. And so she's been docking or making different compounds and docking those virtually with this protein structure. And it's been quite interesting to see how the different compounds do or don't interact with the fungal enzyme. And hopefully that's going to develop further, and there's going to be more, investment in that side of things. And I suppose one of the epic moments, and I suppose in everybody's science life that there is an epic moment, was being invited to be part of the transatlantic yeast gene deletion project. Bit of a mouthful. But the aim of that was, it was set up between a lot of different US labs and our own lab, which was the only UK lab at the time to be invited to join it. So a huge privilege. And the aim was to knock out all the genes in this yeast, Saccharomyces cerevisiae, baker's yeast. And with the aim of trying to work out which ones were essential for life. So essentially, if you knocked out a gene and the yeast died, you could then say it was essential for life. And so we could get to a minimum number of genes and proteins that required for life. And this fed directly into the Human Genome Project, because there were a lot of similarities between the genes that are present in yeast to the ones that are present in our own cells. And so this was a real privilege to be part of something so big and groundbreaking. As I looked back, I could see that the technology has become really quite old-fashioned, although at the time it was innovative and we were all really sort of up for it and really excited by it. But thrillingly, it's been superseded by the more modern technique of CRISPR, and that 2 female scientists this year were awarded the Nobel Prize in Chemistry for this new way of knocking out genes. And I suppose from my own perspective, it's been really exciting to see that being adopted in our research group, and that technique is now being used. So nothing ever stays the same, and that in itself is exciting in science.
You can obviously see that you're passionate about what you do. So I suppose many ways you've already answered this next question, but what makes you get out of bed for research?
Yeah, there was no easy answer to that either, Claire. And day to day, I would say it depends on the stage of a particular project is at. So it's certainly helpful to have more than one project on the go at a time, and that is quite typical in a research lab. And this is because sometimes progress can be painfully slow in one area and much faster or moving more quickly in another. So you've always got something to be working on. And in my experience, it's working as a team to solve the challenges that make it worthwhile. Science is not just one person, or very rarely one person, it's teamwork. and the feeling that you're collectively contributing to knowledge, while being able to work on something that you're really interested in. And I've been so blessed all through my research career to be able to work on something that I am passionate about and that I do enjoy working on. Yeah.
So looking more towards 2020, what does a week of research look like for you now, pre and post lockdown?
Yeah, well, it's been, as we know, a very strange year. So perhaps the biggest difference is the number of Zoom meetings that I've been involved in. I'd only ever had one Zoom before March 2020, and I've actually lost count of how many since. And again, I think context is important. Most of my work is done in meetings with researchers, and not actually in the lab myself. In fact, I think the lab would probably freak out if I decided to put a lab coat on and move in and back to a bench. During COVID, there's been a huge exercise in safety in the lab and how the labs can get up and running again. And thankfully, this now has started, but it's restricted. And I think the impact will be largely seen in the years to come, where concerning publications mostly, I would say, because of the delay in actually doing experiments. So that's probably the biggest impact COVID has had so far. Yeah.
And finally, what makes your research worthwhile?
Yeah, I think mostly it's the people I work with. So I think we have a great team in Swansea, and also in the P450 community more widely. So across Europe, the US and Japan. Over the years, many of these wider contacts have become friends, and I've always felt spurred on, I think, after meeting with them, because they know a lot about P450, and they're enthusiastic about it. And that's always good to talk to other people who are also interested in your pet enzyme as well. Also getting research published, that always is a big buzz around that, because it means it's been acknowledged and deemed worthwhile by other researchers in the field. And so I'm very grateful to be part of that research community.
That's great. Thank you for taking the time today to speak to us. It's great to see your enthusiasm. Thank you very much, Diane.
And thank you for inviting me.